Hello everyone, welcome to my channel Biochemistry Club. Welcome to the second lecture of the series, and today we will discuss how the exergonic and endergonic reactions are coupled. So inside the living organisms, the sum of all metabolic reactions is collectively called as metabolism, and the metabolic reactions can be classified as catabolic reactions or catabolism and anabolic reactions or anabolism. These reactions, they release energy, and anabolic reactions, they demand energy. And why is it so? In catabolic reactions, the complex reactants, they undergo conversion to the simpler products having lower energy status. So a complex reactant, when it will be converted into the simpler product, which requires lower energy, the additional amount of energy will be released. And this is the reason that catabolic reactions, they always proceed with release of delta G or the loss of delta G value, and their delta G values are always negative because reactants, they have much more energy compared to the energy required for the formation of products. So during a particular chemical reaction, they will make products and they will release extra amount of energy. So their delta G values will be negative. Example of such reactions include hydrolysis of proteins, where proteins are complex and when they will be converted into the amino acid, the constituent products are simpler. And this reaction will proceed with release of delta G, release of free energy. Similarly, conversion of glucose to carbon dioxide and water always proceeds with release of energy, which is then used for the formation of ATP. On the other hand, the anabolic reactions, they always demand energy. And why is it so? Because in these type of reactions, the reactants are simpler, reactants have lower energy, and for formation of products, additional amount of energy is required. So if we do not provide this extra amount of energy, the chemical reaction will not take place. So these chemical reactions, they demand energy. So inside the living organisms, there are some couplers, energy couplers, and transporters of reducing equivalents. So when living organisms, they carry out their catabolic reactions during which their bonds are broken down, electrons and hydrogens are being released. So in order to pick up these hydrogen and electrons or reducing equivalents, there are some cofactors, some coenzymes like NAD, NADP, FAD. So these cofactors or coenzymes, they will pick up the reducing equivalents and they will be converted into their reduced form from their oxidized form. Similarly, there are some other carriers which will pick up the energy which is being released during catabolic reactions, and they will be converted into their energy rich forms, ATP. Now, these reducing equivalents and these energy couplers will then be used to provide energy and reducing equivalents to anabolic reactions. So what I want to make clear here is that inside the living organisms, the endergonic reactions are coupled to exergonic reactions. Exergonic reactions release energy and endergonic reactions demand energy. So whenever a chemical reaction is being taking place and it is releasing energy, this energy is then be used to link or to couple another reaction which requires energy. So this is how living organisms manage their endergonic reactions by coupling them with exergonic reactions. So this way, the endergonic reactions gets the energy which they requires to make a reaction possible. So from this, we should keep in mind that delta G energy values of different chemical reactions can be additive. For example, if this is a half reaction in which A is being converted to the B and then B is converting into the C, so the sum of chemical reaction in which A is converted into the C, this reaction will have the delta G value, which will be equal to the sum of these two delta G values. So delta G values of different chemical reactions which are coupled, their values of delta G's are additive. This can be further clarified from an example. Suppose there is a chemical reaction that is the phosphorylation of glucose. So if we see glucose by accepting inorganic phosphate can be converted into glucose 6-phosphate, but this chemical reaction requires 13.8 kilojoule per mole of energy. So this reaction is thermodynamically non-spontaneous, non-favorable. So in order to make this process favorable, another process, which is ATP hydrolysis, is coupled to this process. So hydrolysis of ATP, which is the second reaction, its delta G naught is minus 30.5 kilojoule per mole. Ergonic, the first reaction is endergonic, which requires energy. Second reaction is exergonic, which release energy. So if we add these two reactions, the sum of these two reactions, because these two reactions are now coupled, the sum of these two reactions will be glucose plus ATP into ADP plus glucose 6-phosphate, because inorganic phosphate is present in both sides, reactants as well as product. So it will be cancelled, and this will be the total sum of these two reactions. And if we calculate the delta G value of these reactions, we can add delta G naught 1 and delta G naught 2. And now the combined free energy value will be minus 16.7 kilojoule per mole, which indicates that hydrolysis of ATP by releasing 30.5 kilojoule per mole of energy, it is providing 13.8 kilojoule per mole of energy to this particular reaction and making it favorable. And still, extra 16.7 kilojoule per mole of energy is being released. It means after coupling, after fusion, still this chemical reaction is favorable and spontaneous. So this is the principle of energy coupling inside the living organisms. Now, another important question is why ATP is mostly used as energy coupler? What is the reason that ATP is used as energy coupler in almost all chemical reactions? There are several reasons. There are several factors which makes ATP a good coupling factor. So one of these reasons is the structure of ATP is thermodynamically unstable, and the products inorganic phosphate and ADP are stable. If you see the reaction coordinate diagram, unstable means higher energy state. So ATP is unstable and its hydrolysis products, including ADP and inorganic phosphate, are stable. So whenever a nucleophile will conduct an attack on one of the electrophilic centers of the ATP, because this ATP is unstable, it will immediately breaks its structure. It will immediately convert into its products because it is highly unstable. So you can say that ATP was waiting for some nucleophile to attack on its electrophilic phosphate. And in response to nucleophilic attack, it will immediately breaks its unstable structure and will produce the stable products. So this is the one reason that the products of ATP hydrolysis are more stable. Products are more stable than reactants. And whenever the products are more stable as compared to reactants, this scenario makes that particular reaction a favorable reaction. So hydrolysis of ATP is very favorable. Whenever a nucleophile will conduct an attack from any structure, ATP will immediately break its structure. So this makes it a good energy coupler, because if an energy coupler is not breaking its bond in response to a nucleophile, that means it's not a good energy coupler. It is not breaking its structure. It is not releasing energy. So this is the first reason which makes ATP as an ideal energy coupler. Now, let's move towards the second reason. First one was more stable products. Now, the second one is release from charge repulsion. Release from charge repulsion. If you see the structure of ATP, there are four minus charges. ATP 4 minus. And these similar charges, they repulsion, they create a charge repulsion on the structure of ATP. So once a nucleophile will conduct an attack, for example, on terminal phosphate, ATP will break its bond with the rest of the structure ADP, and this phosphate, it will make the bond with nucleophile. So keep this thing in your mind that on which phosphate the nucleophile will conduct attack, that particular phosphate will break its bond to any other group and it will attack to the nucleophile. So this terminal phosphate will break its bond with the rest of the structure and it will be then attached to the nucleophile, just like this. So after the breakage of this bond, in response to the nucleophile, ATP 4 minus will be converted into the ADP 2 minus because two of the negative charges have been released. So we can say that hydrolysis of ATP will relieve it from the charge repulsion. So again, ATP will preferably break its bond in response to a nucleophile because it wants to get relief from charge repulsion, which is not a favorable thing. Now, the third one is resonance stabilization. Resonance stabilization. As you are seeing here, the hydrolysis product of ATP, the first one is inorganic phosphate and second one is ADP. And you can see here that inside the inorganic phosphate, there is a double bond between phosphorus and oxygen, and this double bond resonates between these other oxygens. So we all know that resonating structures are much more stable. So because of the presence of phenomena of the resonance in the products of ATP hydrolysis, its products becomes more stable and ATP hydrolysis becomes more favorable. So we can say that ATP is an ideal energy coupler, which is ready to break its bond, which is ready to transfer energy to any nucleophile. But the condition is the nucleophile has to conduct an attack on one of its electrophilic centers. So attack is the main point. Once the nucleophile will successfully conduct attack on the internal phosphate of ATPs, it will immediately break its bond because it is ready to break itself in order to get relief from charge repulsion to make products which are more complex, stable as compared to ATP. So this was the first question. Now there is a second question that if you see the status of ATP on this energy chart is representing the energy of hydrolysis of different compounds. There are some low energy compounds, there are some high energy compounds, and you can see the position of ATP is intermediate. There are some low energy compounds as well and some high energy compounds as well. So the question is, an energy coupler should have high free energy of hydrolysis. But if we see the free energy of hydrolysis of ATP, it is minus 30 kilojoule per mole. However, we have some other compounds, 1,3-bisphosphoglycerate, phosphocreatinine, and phosphoenol pyruvate. And if you see the delta G of hydrolysis of these compounds, 1,3-bisphosphoglycerate, phosphocreatine, and phosphoenol pyruvate is far greater than free energy of hydrolysis of ATP. For 1,3-bisphosphoglycerate, the delta G naught is minus 49.3 kilojoule per mole. For phosphocreatinine, the free energy of hydrolysis is minus 43 kilojoule per mole. And for phosphocreatinine, the value is 61.9 kilojoule per mole. So these three values are much higher than free energy of hydrolysis of ATP. So why ATP is an energy coupler? Why not these high energy compounds? Because they are offering more energy of hydrolysis. So answer to this question is an ideal energy coupler should be on intermediate position on energy chart so that it can easily provide energy to low energy molecules. And when its on concentration starts declining, there must be some high energy molecules which will immediately replenish it. They will produce ATP by giving 30 kilojoule per mole energy. But if phosphoenol pyruvate would be an energy coupler, once it would have provided 61.9 kilojoule per mole energy to the low energy compounds, its own production would become a challenge for cell because cell do not have any other molecule to provide more than 61.9 kilojoule per mole energy. So ATP, its intermediate position on energy chart makes it an ideal energy coupler because it can easily transfer energy to low energy compounds and further high energy compounds will replenish its concentration. So its concentration will be maintained inside the cell. So now let's see why some phosphorylated compounds and why some esters have large free energy of hydrolysis. So in phosphorylated compounds, we have two examples, phosphoenol pyruvate and then phosphocreatinine. These two compounds have more or large free energy of hydrolysis. So if you see the structure of 1,3-bisphosphoglycerate, so once this 1,3-bisphosphoglycerate will undergo hydrolysis, this phosphate, which is attached to the carbon number 1, it will be released. And here a carboxylic acid functional group will be established. So the hydrolysis product of 1,3-bisphosphoglycerate is 3-phosphoglycerate. Now, in this 3-phosphoglycerate, there will develop, after ionization, there will be developed a resonance stabilization. And I have previously mentioned that the structures where the products have resonance, these products are more stable as compared to reactants. So we can say that the free energy of hydrolysis of 1,3-bisphosphoglycerate is greater because after hydrolysis, its products are more stable than its own structure. Than its own structure. So this resonance contributes to large free energy change. Now the next structure is hydrolysis of phosphocreatinine. Here, this is the structure of creatinine to which the phosphate group is attached, and there is a bond between nitrogen or amino group and phosphate. So now, when this phosphocreatinine will undergo hydrolysis, again this phosphate group will be released by the addition of water molecule. And once this phosphate group will be released, again this carbon atom will shift its double bond between these two amino groups. So again, there will be a resonance structure which will make this product more stable than reactants. In ATP, there was the same case that its hydrolysis products were stable than ATP. But in the case of 1,3-bisphosphoglycerate and in the case of phosphocreatinine, again the products are more stable than reactants, but energy difference is more. The energy of reactants was greater than energy of ATP. So once they will undergo hydrolysis, they will release more energy. Now the third phosphorylated structure is phosphoenol pyruvate, and again its hydrolysis will release one of its phosphate group. And once this phosphate group will be released, there is a process of tautomerization where the double bond will keep shifting between carbon and oxygen. And this tautomerization makes the products more stable than the reactants. So phosphoenol pyruvate, because of the process of tautomerization, it have more free energy of hydrolysis as compared to ATP. So other than phosphorylated compounds, there are some other high energy compounds to which we call thioesters. So first of all, we must clear that how the thioesters are different from normal esters. So simply, an ester bond is R, C double bond O, O, and then R. This is an ester bond. But if the oxygen atom of ester is replaced with sulfur, we call this ester bond as thioester. So you can see here, in the case of acetyl coenzyme A, this carbonyl group is linked with the sulfur atom of coenzyme A. So this is a thioester. Hydrolysis of thioester will break this bond, and this coenzyme A will be released after hydrolysis, and there will be an acidic group, comes from the water. And again, after ionization, there will be the process of resonance, which will make this thioester more stable, which will make the energy of hydrolysis of this thioester large and negative. So if we compare thioester and simple esters, and we say that why simple ester do not have large free energy change as compared to thioesters, so there is a reason. And what is the reason? The presence of sulfur atom in thioester makes them highly unstable, or they occupy the more energy value they have, and they occupy much higher value at free energy chart. So this is the energy of this thioester energy value. While the presence of oxygen in simple esters do not contribute too much in the energy. So this is the energy value of simple ester. So when thioester will undergo hydrolysis and simple oxygen ester will undergo hydrolysis, they will produce almost similar products of similar energies. But here the difference of energy will be more because the reactant was quite unstable compared to simple esters. So this is the basic rule that why some compounds have more free energy of hydrolysis as compared to ATP. And the reason is, in case of ATP, the difference of energy between reactants and products was not as high. In case of 1,3-bisphosphoglycerate, the difference was comparatively high. In case of phosphocreatinine, again the difference was high. In case of, again the difference was high. In case of phosphoenol pyruvate, the difference was high. In case of thioesters, again the difference was high. So keep this thing in mind that in all these structures, the reactants are unstable and products are stable. That's why these processes have large negative free energy of hydrolysis. But, but some molecules have lower values, some molecules have larger values. And why is it is so? This is because of the difference between the energy values of reactants and products. Okay, so now the next point is ATP transfer its energy by the mechanism of group transfer, not by the hydrolysis. As you can see here, there is a simple single step equation which is representing that glutamate react with amino group, and in this process ATP provides energy by breaking its bond. But this is not a complete reaction. The complete reaction is as follows, which indicates that firstly glutamate interacts with ATP and ATP transfer its phosphate to glutamate. So obviously this oxygen of glutamate will conduct a nucleophilic attack on one of the phosphate of ATP, as I have mentioned earlier here. And if this oxygen is conducting a nucleophilic attack on phosphate, so ATP will transfer its phosphoryl group to this nucleophile, and the remaining structure will be released as ADP. So here you can see that once the oxygen of glutamate will conduct the nucleophilic attack on ATP, phosphoryl group will be transferred to glutamate, and then this amino group, which is the second substrate, it will come and it will replace, it will kick out the phosphate group and it will take the place of phosphate. So ATP do not transfer its energy by simply breaking its bonds, but it transfer its energy by group transfer mechanism. And how many types of groups ATP can transfer? It totally depends upon the phosphate where a nucleophile is conducting attack. So if a nucleophile is conducting an attack on terminal phosphate or the gamma phosphate, outermost phosphate, then this nucleophile is interested in making bond with this phosphate. So definitely, if this phosphate has to make a bond with nucleophile, it has to break its one of the bond with any of the structure. So if this phosphate has to make a bond with nucleophile, it should break its bond with the remaining structure. So here, this bond will be undergo hydrolysis and phosphate group will be transferred to nucleophile. So this is called phosphoryl transfer. And along with phosphoryl group, the remaining structure will be released as AD. So the second scenario is when a nucleophile conducts an attack on beta phosphate. Now, this beta phosphate is linked with gamma phosphate and alpha phosphate. So once the nucleophile will conduct an attack, this beta phosphate will look which one of the bond is comparatively weak or easy to break. So it will break its bond with alpha phosphate, and this complete structure, because it has not broken its bond with the gamma phosphate. So these two phosphate, they still will link, and this beta phosphate will make a bond with nucleophile. So now two phosphates groups has been transferred. So now two phosphate groups have been transferred to nucleophile. So this type of transfer is called pyrophosphoryl group transfer, and remaining structure which will be released along with this is AMP. This one, AMP. Now, there is a third possibility when a nucleophile conducts attack on internal phosphate, alpha phosphate. So now, when this nucleophile will conduct attack on this phosphate will break bond with this structure because now this bond is easy to break. So this complete structure, adenine, ribose, and this phosphate will be transferred to nucleophile. AMP will be transferred to nucleophile, adenyl transfer. And pyrophosphate will be released. So you can compare that the electrophilic center where the nucleophile is attacking will actually decide which type of group will be transferred to the nucleophile and which will be released as a co-substrate. So as I have mentioned earlier that there are some properties of ATP which makes it a good energy coupler, including its unstable nature and relief from charge repulsion and resonance stabilization in its products. So whenever a nucleophile will conduct attack, ATP will spontaneously break its structure and will provide energy to the nucleophile by transferring appropriate groups to the nucleophile. But it doesn't mean that ATP hydrolysis is such a freehand process inside the living systems. Though this process is thermodynamically favorable, but kinetically this process is very slow. And this process will only take place whenever an enzyme will come and allow the ATP to hydrolyze by reducing its activation energy barrier. So, though ATP can transfer energy to other chemical reactions, but this transfer of energy is dependent on availability of enzymes, which will reduce the activation energy barrier of ATP hydrolysis. So please keep this point in your mind that kinetically this process is slow, though thermodynamically it is favorable. So the next topic is types of reactions in metabolism, and there are some common types of reactions which we will see in our next lectures when we will be discussing the pathways including glycolysis, TCA cycle, electron transport chain, and fatty acid metabolism, etc. So here I will give a brief, very brief overview of these reaction types. So the first one is the reactions that make or break carbon-carbon bonds. So there are two different mechanisms through which carbon breaks its bond with the next carbon or with the hydrogen. So for breaking and for making bonds with carbon and hydrogen, these two principles are applicable. So the first mechanism of bond cleavage is homolytic cleavage. And as you can see, the bond between carbon and carbon is a covalent bond where there is a sharing of electron pair. One atom from each bonding atom, one electron from each bonding atom will be contributed for this sharing. So if there will be a homolytic cleavage, it means is the bond will be cleaved from this position and each atom will get its part of electron. So what we will get? We will get radicals because any species containing odd number of unpaired electrons is a radical. So radical formation is the result of homolytic cleavage of carbon-carbon and carbon-hydrogen bonds. In case of hydrogen, we will get one hydrogen atom because hydrogen contains normally one electron and one proton. The second mechanism is heterolytic cleavage, where the bond cleavage is not symmetrical. It is asymmetrical, which means its bond is cleaved from any extreme position, close to the carbon or close to the hydrogen. For example, if bond is cleaved close to the hydrogen, the carbon atom will get both electrons and it will be a carbanion, and hydrogen will not get any electron and it will be a proton. And in other case, if the bond is being cleaved close to the carbon, in this case, carbon will not get any electron and it will be a carbocation, and hydrogen atom by gaining two electrons will become hydride. Now, in case of carbon-carbon, when unequal cleavage will take place, one of the carbon atom will be carbanion and the second carbon atom will be cation. So the formation of carbanion, carbocation, hydride, and proton is the result of heterolytic cleavage, and formation of free radical is a result of homolytic cleavage. Now, if we see the reverse of this process, it means one carbon radical requires second carbon radical if they want to make a carbon-carbon bond. And for carbon-hydrogen bond, carbon radical will be demanding a hydrogen atom. Similarly, if there is a carbocation, the next must be carbocation, and they will make carbon-carbon. And carbanion and carbocation, if they want to make bond with hydrogen, they require proton or hydride, respectively. The second principle is in biochemical reactions, there are some species which are rich in electrons and some species which are deficit in electrons. And these two species, they can make bonds. If there is a nucleophile and it conducts an attack on electrophilic center, these two species can make bond because this how they can manage or they can sink their electrons to a common pool. So these are the examples of some common nucleophiles, and these are the examples of some common electrophiles inside the living organism. So we will discuss it in more detail when we will be referring to an example where some nucleophiles are conducting attack on electrophiles. Okay. Now the most important point is the significance of two important groups, and I will refer to this point during my discussion on glycolysis and TCA cycle because here I will refer to the significance of carbonyl functional group in the cleavage of carbon-carbon bond. So the carbonyl and amine groups play a very important role in metabolic reactions. If we talk about the carbonyl functional group, this is the group where the carbon is attached with electronegative oxygen through a double bond. And oxygen being an electronegative element, can sink electron towards itself, developing a partial negative charge on it and partial positive charge on the carbon. This makes this carbon a good electrophile. Now, the attachment of this carbonyl group to a carbocation stabilizes this carbocation. Now, the attachment of this carbonyl group to any carbanion stabilizes it. Otherwise, this carbanion cannot make a bond. It is highly unstable, highly reactive. So the carbonyl functional group can sink some of the electrons from this carbanion and can stabilize it. The same role can also be played by the amine group. As you can see here, this amine group can sink some of the electrons from carbanion and can stabilize it. So these two groups are very important in metabolic reactions. And the significance of carbonyl group, it can be evident in three reaction types: aldol condensation, Claisian condensation, and decarboxylation, where these two reactions, they involve carbon-carbon bond formation, and the third one requires the breakage of one carbon-carbon bond. So here you can see in aldol condensation, a carbonyl functional group attached to a carbanion, and then this carbanion is making a carbon-carbon bond with the next carbonyl. So this type of condensation or this attack of this carbanion on this electrophilic center is possible just because of the presence of this carbonyl functional group just close to this. Similarly, in case of Claisian condensation, again, the presence of carbonyl functional group close to the carbanion stabilizes it and makes its nucleophilic attack on the next electrophilic center possible. So now, this nucleophile can make a bond with this electrophile. And again, this condensation is possible because of the presence of this carbonyl functional group close to the carbanion. So here you can see the involvement of carbonyl functional group in making carbon-carbon bonds. Now, reverse can be seen here. The carbonyl functional group within a structure can make the next carbon-carbon bond, adjacent carbon-carbon bond. For example, if this is carbon number 1, so the presence of this carbonyl functional group have the potential to make the bond between carbon 2 and carbon 3 weak. Adjacent carbon-carbon bond can become weak in the presence of this carbonyl functional group. So what will happen when some nucleophile will conduct attack, or when a substituent want to take the position of this carbon dioxide? This will be released as a leaving group. So this decarboxylation, this release of carbon dioxide from this structure has become possible because of the presence of the carbonyl functional group behind this. The second reaction types are internal rearrangements, and here you can see either a functional group or any other group can be transferred from one position to another. For example, the enzymes isomerases, they have potential to transfer the carbonyl functional group from carbon number 1 of the glucose to carbon number 2 of the fructose. So such type of reactions where within a structure, a molecule is being transferred, a group is being transferred from one position to another position, these type of structures, they comes under the heading of internal rearrangements, isomerization, etc. The third type is group transfer reaction, and we have already discussed this type while we were discussing the ATP as a energy coupler. And here you can see that this is the first reaction of glycolysis, where the glucose, this is the first reaction of glycolysis where the glucose is converted into the glucose 6-phosphate. So for this conversion, what glucose does, the carbon number 6 of the glucose, where the OH group is attached, and oxygen of this OH group conducts a nucleophilic attack on the phosphate, terminal phosphate of ATP. And as I have discussed earlier, that once a nucleophile conducts an attack on electrophilic center, that electrophilic center tends to make bonds with the nucleophile by breaking its bonds with the other structures of the ATP. So this phosphoryl group will break its bond with the rest of the structure and will release ADP, and phosphate group will be transferred to oxygen of carbon number 6, oxygen of the OH of carbon number 6 of the glucose. So glucose will be converted to glucose 6-phosphate by this type of reaction, which is a group transfer reaction because ATP is transferring its group to the glucose. The fourth type of reactions are free radical reactions. And as we have discussed earlier, that free radicals they form during homolytic cleavage of bonds. So there are many chemical reactions in metabolism where the homolytic cleavage converts a structure into the free radical or a carbon or any other group to the free radical, and then this free radical can create some further free radicals in these structures. So the formation of heme group is one of this example. As you can see here, this free radical will grab the proton from here, and by grabbing the proton, it will create a free radical in the structure, which will then further be stabilized by breaking of this bond, this one. The final type of reactions is oxidation reduction reaction, and please keep this thing in your mind clear that these two reaction types are coupled. Oxidation is any reaction where electrons are being released, where hydrogen is being released, or where oxygen is being added. Similarly, reduction is any process where electrons are being added, hydrogens are being added, or oxygen is being removed. But a particular species will only undergo the oxidation when there is a species which is willing to accept its electron or hydrogen. For example, if there is a substrate A and it has hydrogen and there is a substrate B, A will only release its electron when B is willing to accept these electrons. And how we can decide that B is willing to accept its electron? It all depends upon their strength, their affinity for electrons, which can be calculated by their reduction potential. So any molecule having high reduction potential can grab the electron from another molecule. So if B has high reduction potential, when this B will come close to the A, now A cannot hold this electron. This electron will be released and B will grab it. So the species having higher reduction potential will always be reduced, and species having low reduction potential will always be oxidized. And this competition of reduction potential will only develop when these two species will come close to each other. So this is a basic point, very basic point. Now, keeping this point in your mind clear, we will discuss some of the example of biological oxidation reduction reactions. Inside the living organisms, mostly the oxidation reduction reactions, they involve removal of hydrogen or removal of electron. And one of the most common type of oxidation reduction reactions in biological systems includes dehydrogenation, which means that if a species is going to be oxidized, it will release a molecular hydrogen, two hydrogen atoms in the process of dehydrogenation. Similarly, if there is another species which is willing to accept this electron, it will accept the molecular hydrogen. So the dehydrogenation is any process where the species being oxidized will release two hydrogen atoms, and the species being reduced is willing to accept these two hydrogens. For example, if there is a species A, this A will release two hydrogen atoms or one complete molecule of hydrogen, molecular hydrogen, then the mechanism of oxidation will be hydrogenation. And if this species is being oxidized by releasing molecular hydrogen or hydrogen, there must be another species which is willing to accept these hydrogen and it will undergo the reduction because these two processes are coupled. The most common type of biological oxidation reduction reactions is dehydrogenation. And in order to clear this, we will take the example of lactate dehydrogenase, which is an enzyme which have the potential to convert lactate to pyruvate. Here you can see that this enzyme is dehydrogenase, which means it is willing to it. Here you can see the name of enzyme is lactate dehydrogenase, which means it will carry out the mechanism of dehydrogenation and it will remove a complete molecular hydrogen from the lactate. So one hydrogen from here, one hydrogen from here, two hydrogen atoms, because one hydrogen atom is a combination of one proton and one electron. Two hydrogen atoms mean two protons and two electrons will be released. Two hydrogen atom or molecular hydrogen will be released. And this carbon will make double bond with oxygen and the lactate will be converted into the pyruvate. This is the oxidation of lactate. Lactate has removed hydrogen and it has oxidized to pyruvate. But where these hydrogen and electrons will go now? So the enzyme lactate dehydrogenase does not work alone. It always requires a cofactor, which is NAD+. And once enzyme will release a molecular hydrogen from the substrate, this NAD will pick this molecular hydrogen and will accept hydride ion from it and it will be reduced. So if there is an oxidation, there must be a reduction. So one substrate is being oxidized and there is a second species which is being reduced. So this is the example of oxidation and reduction reaction. Now, biological oxidation reduction reactions can be quantified or their free energy values can be calculated by the formula delta G naught is equal to minus NF delta E, where N is the number of electrons being transferred. For example, if we can see here the two electrons are being transferred from lactate to NAD+. F is the Faraday constant having a constant value of 96.5 kilojoule per volt. And delta E naught, this is the difference in reduction potential which we have to calculate. So how we can calculate delta E naught? It is equal to reduction potential of acceptor minus reduction potential of donor. Or in other words, reduction potential of species being reduced or reduction potential of species being oxidized. So in this previous case, lactate is being oxidized. So if I have reduction potential of lactate, NAD plus is being reduced. And if I have reduction potential of NAD plus, I can put these values in this equation and I can calculate the value of delta E naught. Now the question is, from where we can get the reduction potential value? So these values are constant, which have already been calculated. So just like standard hydrogen electrode, we have reduction potential of different half reactions already calculated. And we can take these values in order to calculate the delta E naught. So let's solve an example to get a better understanding of reduction potential and calculation of delta E. So let's solve an example to get a better understanding on how to calculate the free energy change of oxidation reduction reaction. So this is an example where we have been asked to calculate delta G naught for the reaction in which acetaldehyde is reduced by biological electron carrier NADH. From this equation, we are getting an idea that NADH is being oxidized because its reduced form is being oxidized in product and acetaldehyde is being reduced. So this is oxidation, this species being oxidized, and this is reduction, this species being reduced. And you can see here, we have also given the half reactions showing the reduction of acetaldehyde and reduction of NAD+. But we know here NAD+ is not being reduced, it is being oxidized. So what we will do, we will take the values of reduction potential and we will calculate delta E naught is equal to E naught of reduction minus E naught of oxidation. So in this case, acetaldehyde is being reduced. So we will put minus 0.197 here and then minus NAD is being oxidized. So we will put its value again. Now the sign or the values will not be changed, whether the species being oxidized or reduced, but only the arrangements will be changed. So if we take delta E naught, it will be 0.123 volts. And once we have delta E naught, if we get the positive value of delta E naught, it means reaction is possible. So by putting the value minus NF delta E naught, here the two electrons are being transferred, as you can see here. So the number of electrons will be 2. Faraday constant is 96.5 kilojoule per volt. And then the delta E naught, which is equal to 0.123 volt. Here kilojoule per volt and volt will be cancelled. And what we will get minus 23.7 kilojoule per volt. And this minus sign is indicating that this reaction is possible, this reaction is favorable. So this is how we can calculate the free energy value of an oxidation reduction reaction. Now, as you have seen in the case of lactate dehydrogenase, that mostly the enzymes dehydrogenases, they are linked with coenzymes. And these coenzymes are FAD, NAD, or NADP. So when this enzyme converts a species, for example, species A, to its oxidized form and releases 2 electrons, these coenzymes can pick those electrons and they can be converted into their oxidized form they can be converted into their reduced form from their oxidized form. So now we will see that how these coenzymes or how these electron carriers, they pick electron and how they are different with each. So this is an example of nicotinamide adenine dinucleotide, which is normally used as a coenzyme for variety of enzymes, including lactate dehydrogenase, pyruvate dehydrogenase, and many other dehydrogenases. So when dehydrogenases, they oxidize a substrate and they remove a molecular hydrogen, including 2 electrons and 2 protons. This nicotinamide adenine dinucleotide can accept a hydride ion from this molecular hydrogen. Now, what is hydride ion? One hydrogen atom is a combination of 1 proton and 1 electron. Second hydrogen atom is a combination of second proton and second electron. And this molecular hydrogen, these 2 hydrogen atoms are being released by enzyme. Now, NAD will accept 1 proton and 2 electrons. A simple hydrogen atom contains 1 proton and 1 electron. But when there will be an additional electron, this will not be an hydrogen atom, this will be an hydride ion. So NAD out of these 2 protons and these 2 electrons can accept 1 proton and 2 electrons in the form of hydroiodine and can incorporate this hydroiodine into its structure. And remaining one proton, it will be written as. So this is the reason whenever an NAD is converted to its oxidized form, we write NADH plus proton, which is an indication that two electrons and one proton is the part of its structure and the other proton is released in medium. Now, the second coenzyme, the same is true for NADP. So NADP, it will accept molecular hydrogen, it will incorporate hydroiodine into its structure and will release proton as it is. Now the next example is FAD. And what FAD do? FAD can accept electron in the form of hydrogen atom. So one electron and one proton. So suppose there is an enzyme, say succinate dehydrogenase, to which this FAD is covalently attached. So when this enzyme converts succinate to fumarate during TCA cycle, it releases a molecular hydrogen, which means two hydrogen atom comprising of two protons and two electrons. So now, what FAD will do? FAD can accept one complete hydrogen atom, means one proton and one electron. It can accept one hydrogen atom and can be converted into the FADH, which is a partially reduced form. But still it has potential to accept second hydrogen atom containing proton and electron and can be fully reduced to FADH2. So NAD can accept hydrogen in the form of hydride ion. So NAD can accept electrons in the form of hydride ion, which means two electrons and one proton. But FAD can accept electrons in the form of hydrogen atom, one electron and one proton. So that's why FAD can completely accept molecular hydrogen and it can be converted from FAD to FADH2, accepting both hydrogen atom into its structure. As you can see, one hydrogen atom and second hydrogen atom. But NAD can accept only hydride ion. It accepts one proton and two electrons and release second proton as it is. So this is the last of bioenergetics and biochemical reaction types. If you have any question, you can ask in comment section. Stay tuned because my next lecture will be on glycolysis.